All right, we start with a sound check. Can you hear me in the back row? Can you hear me in the back row? Excellent. Great. So we've come to the end of Chris's lectures, and they'll be back to me again, if I can get it to work. There we go. And before we actually do the science things, which are more interesting, we have a little bit of course matter to sort of keep on track of. Just a reminder, it's in the syllabus, but you might not have looked at it lately. Today and Friday, we'll be finishing up Chapter 19, which deals with transcription and some topics about RNA that I thought might be of interest. We may not get clear through them. If not, don't worry, you won't be expected to know them. Monday and Wednesday, we'll be talking about Chapter 20, the last one in the book, which is about protein synthesis. And then Friday is the 4th hour exam. Kind of close, but on the other hand, sometimes things end up close. Susan wanted me to let you know that she has arranged for a room for a review session on Wednesday. So that would be Wednesday the 25th, and we'll let you know what the room is on it. It'll be in here. Wednesday the 25th in here, 6 again? Okay. So the last hour exam will cover through the end of Chapter 20. The last week of lectures will be a review of the whole course to help you put your thoughts together, refresh your mind, and so on. And then the final exam is May 7th. We are split between 2 rooms, and so I will be announcing which room you ought to go to. Some people will come here, and some people will go, I think, across to Bowen. But to just keep that in mind. And the final exam is a comprehensive final, 100 points over all the semester. Chance to sort of put different parts together. Any questions about these course matters before I move into the interesting part? Yes? Yes. Okay, there will not be a study group on the 25th with Angie then. Okay. All right. So let's start talking about RNA, a very interesting molecule. DNA is, of course, the center of things. It's what genes are made out of. But I once outraged the editor of Cell by saying that DNA is the dead hand of the past. DNA by itself can do essentially nothing. And so DNA depends on there being some way to get that information out of the gene into real life. And RNA is an important part of that, as we're going to see. Not the entire, but a good big part of it. And one of the things that's allowed us to probe how that works is, oddly enough, this poison that comes from a mushroom called Amanita phalloides. Nice picture of it in your book here. It's also called death cap because it does such a good job of killing things. Amanita phalloides produces alpha-amanitin, which is a peptide antibiotic that will inhibit RNA polymerase. And so it was a tool in the early days to start to understand how RNA polymerases work. Once you stop making new RNA, protein synthesis will fall off gradually as the old RNA gets destroyed. Interestingly enough, although alpha-amanitin stops messenger RNA synthesis, it is not the alpha-amanitin that kills you. It's the phalloidin in it that kills you because it makes your liver look like a pincushion or an urchin. The tasty bit is from a story a friend of mine told me where he was on Cape Cod and saw someone picking Amanita phalloides and felt morally bound to stop and ask the man, have you ever gathered mushrooms before? And he said, yes. Have you picked this mushroom before? Oh, yes, but you have to know how to cook it. Oh? He says, yeah, you slice it thin, you soak it in cold water. The water will turn black. You throw that away. You keep changing the water until it comes off clear, and then you fry it in butter. So apparently, Amanita, alpha-amanitin and phalloidin are both water soluble. So I wouldn't try it. But there you have it. All right. So we're going to start talking about transcription. And transcription, or transcribing something, is copying the information that's in the linear sequence of DNA into a more active form, which is the RNA. And clearly, this is a point at which you have to be picky. When we talked about replication, it's crucial that cells replicate all of their DNA. In that case, the challenge is to make sure you got all of it. In the case of transcription, the challenge is to only do some of it. And a lot of the focus in transcription is making sure that the right gene is transcribed at the right time and the wrong gene is not transcribed. And so there's going to be layers of regulation that control that gene expression. A transcript is a piece of RNA that comes off of a copying episode off of DNA, off of a DNA template. And it's interesting, the kind of interrelationship between that and what we often think of as a gene. In the case of messenger RNAs, they're coding for protein. That transcript carries the sequence that'll make a protein. There are a number of RNA molecules that are themselves the thing that has an effect. So ribosomal RNA, transfer RNA, small nuclear RNAs. These are RNAs which by themselves do something. And so in the absence of translation, those are the thing that's needed. And the phenotype would come from the failure of those RNAs to be that RNA. We're going to look at processing. A lot of the transcripts end up being processed before they're useful. There are areas of DNA that are not transcribed. That is, they don't make any transcript, they don't make any RNA, they don't code for any protein, and yet they can control other things. And so they can have a phenotype, an inheritable phenotype, a mutable phenotype, and that is behaving along the classical definition of a gene. And so there's this kind of overlapping, most genes have transcripts, most transcripts belong to genes, but it's not a perfect matching. You can have non-transcribed DNAs with a phenotype, and you can have transcripts where there's no detectable phenotype. So it's not a perfect one-to-one matching. Okay. So how many are we talking about? We now have the complete genomes for a number of organisms. The human genome was one of the first ones that was done. And human beings, surprisingly, only have 30 to 35,000 genes. Before the human genome was completed, the estimates were twofold or more higher. But when we look at those 30,000 or so genes, they comprise only 1% of our DNA. So most of our human DNA is not genes. It's DNA that's stuff. In us, in more complicated vertebrates and more complicated animals, those genes tend to have introns. That is, there's a part of the RNA that's present in the initial transcript, but that has to be processed out to make a mature RNA molecule. And we're going to talk more about that. There are a number of things that are called pseudogenes, where you can, so like the ones that are marked in orange here, have aspects that look like another gene, but they might be missing the start site, for instance, or they might have a premature termination. So these are things that probably began life as a gene, but have been mutated into something that's no longer functional. Genome size, we're going to see, can vary remarkably, and not necessarily with the kinds of complexity you think of. There are frogs that have 5 times as much DNA as we have. And that probably the more useful number, rather than simple size, is the number of regulatory genes, because it's the regulatory genes that allow you to do things in new and interesting and complicated ways. Okay. Now, Carissa talked about how replication happened in replication factories, that there were locations in the nucleus that had the machinery that was needed in order to replicate DNA. And it turns out that transcription is the same way, but they're different factories. And so it's possible to light these up with different markers. And in this case, they've used a green labeled deoxynucleotide, and in this case, a red labeled ribonucleotide. And in this case, it's been incorporated into DNA molecules. And in this case, it's been incorporated into RNA molecules. And so you can do the overlay of the two, and you can see that the two rarely coincide. So that the nucleus is using different bits and pieces, either for replication or for transcription. And one of the major challenges in these factories is going to be some way of opening up the DNA and making an RNA molecule without getting hopelessly tangled up. And so looking at it in a very broad sense, this is the challenge that we're going to be faced with. There has to be a polymerase molecule that can interact with the DNA, that can open up enough area that you can make a complementary stretch to it, and still have the RNA molecule escape without getting tangled into the DNA also. So this is the overall demands that are placed on an RNA polymerase molecule. A little different than the DNA polymerases. Similar, but somewhat different. So how does the polymerase know what to do? Transcription begins at what are called promoters. And a promoter is defined as the place where transcription begins, and so that's necessarily true. In prokaryotes, we have two sequences that tend to show you where the promoter is relative to other sequences, or where the polymerase will start. So there's a sequence tatat at -10, and -10 means 10 nucleotides upstream of where the polymerase starts. And then a second one, TTGACA, at about -35. You don't need to memorize the sequences, but there are two of these, and they have a fairly specific spacing relative to each other and relative to the start site. But that's just the DNA sequence. It's what is it about the polymerase that can interact with those sequences? Well, the E. coli polymerase is a multimer. It's an alpha-2 beta prime complex, and that's the polymerase per se, so to speak, and then a sigma. And sigma is the additional protein on any given polymerase complex that targets that particular complex to one promoter over another. So there are multiple copies or multiple versions of sigma that are going to be targeting to different kinds of promoters. Okay. Eukaryotic promoters are a little more complicated. There is usually what's called a TATA box. Doesn't that sound sort of like the TATAT of prokaryotes? That's about 24 nucleotides upstream of the start site. But apart from this TATA box, there is an immense variety of what else upstream is involved in telling the polymerase this is where to begin. There are entire courses taught solely about what the upstream sequences are that control transcription. So we're not going to be able to exhaust that particular universe. Eukaryotic polymerases don't use sigma. They don't have any homologue to that. But instead, there are a large variety of transcription factors, and these are proteins that can engage in protein-protein or protein-DNA contacts that create a pattern specific to that gene to turn on transcription at that place at that time. It's really kind of amazing that the polymerase can find the promoters. I already said that the genes are only 1% of the DNA, and the promoters are a tiny fraction of the genes. So somehow or other, the polymerases have to find those promoters and interact with them, because, as Chris has already told you, there are histones that the DNA is wound around. There are chromatin proteins that help to pack it into the solenoid and so forth. A lot of protein on it. And it appears more and more the case that the kinds of protein and the condition of that protein are part of what help a polymerase know where to go. So, for example, Chris has already alluded to the fact that there is a portion of the interphase nucleus called heterochromatin. And these are portions of the genome that are packed in a very dense way. They stain very darkly. And in those regions, there are not very many genes transcribed. So there seems to be a relationship between the tightness of the packing and the accessibility for transcription. The other major component of the genome is called euchromatin. Eu is the Greek thing for good or true or whatever. And in the euchromatin, we know that it's more opened up because it's accessible to attack by things. It has different chemical properties. Carissa mentioned that histones interact differently with DNA depending on whether or not they're acetylated, and we find more of the acetylated histones in the euchromatin area. And this is reminding you here of what an acetyl group can do to lysine. So this would normally have a positive charge on it, and that positive charge would like to interact with the negative charge of the phosphates. But when you acetylate it, you get rid of that positive charge and you let it peel away and open up off of the DNA. And there's growing interest in the extent to which the modifications on the histones themselves provide part of the information for selecting which factors are going to come in, and then the factors interact further with the polymerase. Okay. Eukaryotic transcription is complicated, and it has both the polymerase itself, which is the actual enzyme that can actually make RNA. It has what are called general transcription factors, and these are ones that are going to be part of getting transcription started off of pretty much any eukaryotic promoter. And then in a bit we'll see something about specific transcription factors that help to target specific genes over other genes. And so polymerase II, which is the one that can transcribe messenger RNAs, needs these different transcription factors. They're called TF for transcription factor. So transcription factor 2A, B, D, E, F, and H. TF2D is worth paying a little bit of extra attention to because TF2D has a especially interesting property to it. It binds to that TATA box. This is called the TATA binding protein, was one of its names for it before they knew that it was part of the transcription complex. And then TF2D has with it also proteins hanging off of it, TAFs for TATA binding protein associated factors. And so this is a complex that's going to help identify where the start site is. You know, if you know where the TATA box is, you're getting an idea of where the start site is. And if you look at this physically, you can see that the TATA binding protein can wrap itself around or over the axis or the helix of the DNA, and as it binds, it sort of kinks it a bit to sort of help start the opening up process. The TAFs, the TATA binding associated factors, are not identical from one complex to another. The different kinds of TAFs are going to be part of the way in which specificity is arrived at. And some of those things can actually recognize sequences of DNA. And we're going to see in a little bit the structure remind you of the structure of DNA so you can see why things can bind into or along the structure of the DNA. Another general transcription factor is TAF250, and I like to think of it as the Swiss Army knife protein. This is a protein that has multiple activities. It has a kinase activity. It can acetylate histones, so it's a histone acetylase, acetyltransferase. It can add ubiquitin to histone H1 to target it for proteolysis. Now, ubiquitin, we're going to hear a little bit more about, I think, in the next chapter. It's a universal tag that says to the cell, eat up this protein, it's ready to be degraded. And so what this complex of activities is you can make reversible modifications that will turn things on and off. You can modify the histones to change the accessibility of the DNA, and you can target H1 to be degraded, and that also will help to open up the DNA and make it more accessible. Some of these proteins have regions called bromodomains, which can bind acetyllysine. So here's a ribbon diagram of it, which is looks very open and so forth. And then when you do a space filling diagram, you can see that there's this little place here where you can tuck up that acetyllysine and you sort of pull on it and use that as part of your anchor. So as the complex is sitting there on the DNA, more and more of the general transcription factors can pile on top of it. It's as if each one creates a new template for something else to interact with. And so here you can see one that has the TATA binding protein bound to it, TF2B and TF2A. So you can see how kinked that DNA is because of TATA binding protein. TF2H, they're all TF2s, you notice. TF2H has helicase activity, so that one has a handy name. And as you can imagine, the helicase activity is needed to open up that double helix. Otherwise, you wouldn't be able to reach in and see what the nucleotide bases were inside of it. And so this region that's been opened up is called a transcription bubble, where the DNA has bubbled out. And so once you get this complex all assembled, then that provides a place that polymerase II can dock and start to interact with the promoter directly. Any questions so far? What we've talked about so far are all local things. We talked about the TATA box being, roughly 25 bases from the start site, and these different transcription factors binding there and the polymerase binding there, very local kinds of control. But that is by no means the only kind of control that eukaryotic cells have, and in fact, may even be the least, I shouldn't say the least important, but that there are other things that are equally important. And two of the groups of things that are talked about are enhancers and silencers. And the idea behind enhancers and silencers is that geneticists noticed that there were things that could change the expression of a gene that weren't very close to it. That in fact didn't themselves make anything, but they changed the expression of other things. And so an enhancer is a region of DNA that can increase the expression of genes near it without itself having made a product. So the enhancer is the sequence in the DNA. And a silencer, by contrast, is the other way around. Silencers can turn off genes in their region and create what are called position effects. And this gives you some of the kinds of things that probably are acting in the ways in which these action at a distance genes can work. So here we have a stretch of DNA with an enhancer at a distance from the promoter. This could be tens of thousands of base pairs. It can be a very long distance. And if an activator protein binds at the enhancer and the general transcription factors are binding at the promoter, this can then loop around physically. So now you see the enhancer is physically in contact with the rest of that complex. There's a mediator protein in between holding those two things together. So part of what we think is happening with both enhancers and silencers is the creation of higher order structures in the DNA, where loops can be created that either open up the rest of the DNA or shut down the rest of the DNA in various complicated ways. This particular model is showing it as actually physically interacting at that spot. There are others that would suggest that for some of them, the entire region between the enhancer and, say, the next enhancer is more available for transcription. This is showing you this enhancer with just the one promoter. As you might guess from my comment just now, if there are genes inside of this loop, they might all be influenced by one enhancer. So one enhancer can control several other genes. So the range 10 to the 9th fold, that if you consider how much an enhancer can turn things up and how down a silencer can turn them, overall that gives a range of expression of 10 to the 9th. So this is a huge range for an organism to be able to work from. It gives it great flexibility. Okay. Now, one of the striking things that can control gene expression, and we talked about this a little bit in integration and metabolism, is hormones. And the hormones in the body have a number of different ways of seeing to it that transcription is specific in some cells and not in others. There are some hormones that simply diffuse through the membrane. And when you look at the structures of these, it sort of makes sense. So steroid hormones, retinoic acid, the structures would suggest to you that they could simply diffuse through. The thyroxine is probably carried as if it were an amino acid. But they get through without any great difficulty. And then inside the cell bind the receptor, which then is able to bind to a promoter. For some of these, there is reason to think that that receptor is out in the cytoplasm when it's empty. And that when the hormone binds to it, it then moves into the nucleus and binds to the DNA. So it's not necessarily the case that it was rattling around loose in the nucleus and suddenly got sticky. More likely that it was held out in the cytoplasm and became able to bind and move into the nucleus. We also talked about hormones that could induce a signaling cascade with second messengers and amplification along the way and so forth. That could turn on a series of kinases that would amplify the signal. Obviously, in that case, that hormone is staying at the outside of the cell. So the hormone itself can't be binding to the protein that's going to bind to the DNA. But some of those hormones work through what are called STAT proteins, signal, transducer, and activator of transcription. STAT, of course, means right away, so someone was having fun with their acronyms here. One of the ways in which these things can work is that when the appropriate cascade has been activated, they can phosphorylate the STAT proteins, and then when they are phosphorylated, the STAT proteins can dimerize, and that dimer can bend DNA dramatically. And so you can see here the DNA in the center of the STAT protein, which is holding on to it and able to kink it. So how can we coordinate more than one thing? It's going to be the case quite often that you want this set of genes to all be expressed at a particular time. Not just this gene needs to go on, but these 15 genes need to go at the same time. And so one part of controlling transcription is coordinating things so that a set of things can have the same expression. And this is handled a couple of different ways. In the prokaryotes, those things that pretty much don't have nuclei, many genes are controlled through operons. And this is a made-up word that means a thing that operates, that does the work. And the overall look of it is, for example, here's the tryptophan operon. There are a number of what are called structural genes. So these are genes that code for actual proteins that are going to produce enzyme activity. And then you have a promoter that can turn on or off. Why is that saying little time hourglass? That will determine whether or not that region is transcribed. And when that promoter is active, the polymerase will read clear through the whole region. It'll make one great big messenger RNA. We call that a polycistronic messenger RNA, so that the entire set of structural genes is encoded in one piece of RNA. And clearly then you're going to have coordination of expression. In the case of tryptophan, you only want to invest, look at, there's 6 structural genes here. You would only want to invest your effort in making those structural genes if you didn't have tryptophan around. So this is definitely a case of something that you want to turn off or turn on depending on the environment. And the way this particular one works is that if tryptophan is around, it will bind to a repressor protein, and then the repressor protein binds to the promoter, and then that turns off transcription. So this is a repressible gene. It's on unless it's repressed. Another example of an operon is the lac operon. This has also had a lot of attention given to it. And again, it shows this ability to use a region of DNA in a couple of interesting ways to coordinate the behavior. Here we have the 3 structural genes of the lac operon, Z, Y, and A. These are all involved one way or another in the metabolism of lactose. And lactose, you may remember, is a disaccharide that is not useful by itself until it's split into a glucose and a galactose molecule. And so what you see here in the lac-operon is a permease that lets it get in, a beta-galactosidase that splits it into two enzymes, and a transacetylase. The transacetylase was a mystery for a long time. It looks like it may protect the cell from non-hydrolyzable sugars, but we don't completely understand what it does. There's also a structural gene called I, which makes a repressor. And this repressor will sit on top of the operator and block transcription. So this is its ground state, if you like. Its default state is to be repressed. That repressor protein is always being made, and it will always bind to the operator unless the inducer is present. And the inducer is allolactose, which is a slightly rearranged version of lactose. And if allolactose is present, it will bind to the receptor, to the repressor, sorry, and the repressor will fall off the operator site and make the promoter now accessible and permit transcription through the lac-operon proper. So slightly different strategies. Here, the cell must be betting that most of the time you're going to need tryptophan, and most of the time you're not going to have lactose around. It's making sort of guesses as to what's the most likely state. But in one case, you can repress what's going on otherwise. In this case, you can induce something that won't happen unless you do something active. The lac-operon is of interest in part because it has been the target of a lot of structural work, and we know how it works as mechanistically better than many other things because it's been known for so long. And this is a picture of the protein, the lac repressor. And it is a dimer of dimers. So here is one of the dimers, and here's another dimer. So this spaghetti pile here constitutes one complete lac repressor. And it's kind of like a pincher, like the tongs you use at a barbecue to grab things. And what it does is it grabs onto the DNA and loops it. And so you can see that more dramatically over here. So this is the repressor here, and it's grabbed onto this bit of double helix and that bit of double helix and just tied it in a loop like that. And so in that situation, you can see the RNA polymerase bound, but it's not going to go anywhere as long as that repressor is holding on to the DNA and keeping it in a loop. What this means is that you can have extremely rapid response once the inducer is present. That when this thing falls off, the polymerase is already there at the promoter and can start transcribing the structural genes immediately. So a very clever approach to things. It's also showing this kind of higher order structure as part of gene regulation, even in prokaryotes. So this loop of stuff is, if you like, tertiary structure for DNA because it's over and beyond just the double helical part that's the standard structure. We notice a number of recurring themes among DNA binding proteins that are involved in gene regulation. And this is looking at several of them. The lambda repressor, which we just talked about. Actually, we didn't talk about the lambda repressor. We talked about the the lactose one. But anyway, lambda repressor is another important repressor. Has what's called a helix turn helix. And this is a motif that is seen in many, many DNA binding proteins. And so you have a helix turn and a helix, and that can kind of tuck itself into the major groove. Another strategy are what are called leucine zippers. You may remember back in our talking about protein structure that they're sometimes coiled coils that you can have. Here is one alpha helix here in the gray. Here's another alpha helix in the red. And those two alpha helices can coil around each other in a coiled coil, like the tail of myosin or like keratins and so on. And they might be the same protein and they might be different proteins, okay, depending on the particular system we're talking about. And these leucine zippers then also bind here in the major groove. Okay. A third motif that we see commonly is the zinc finger. This is a very common one in transcription factors. You might remember that the zinc finger is one of the few examples of something where the zinc is structurally required for that tertiary structure to form. It also binds in the major groove. There's a theme that's emerging here. And you might remember back to when we were talking about the structure of DNA that the base pairs, the Watson-Crick base pairs, did not exhaust all the possible hydrogen bonding opportunities, that all the base pairs had some hydrogen bonding opportunities sticking off to the side, different ones for different base pairs. And so what we presume is happening here is that these proteins are reading the sequence in this major groove, that there's a pattern of potential hydrogen bonds, like a pattern of dimples and valleys, and it can fit into that to select that sequence and not something else. Any questions about this before we go on? Okay. Now, in eukaryotes, there are a lot of RNA polymerases. We are blessed with many different ones. And they do different kinds of things. When we talked about the TF2D and the TFH and so forth, that was TF2, meaning transcription factors for polymerase 2, which implies there's other polymerases, which there are. Polymerase 1 is the one that transcribes ribosomal genes and makes ribosomal RNAs. That's obviously something where you don't have a lot of things to select from. You might have a volume control, if you like, but you don't have different ribosomes that you make. Pol2, as I mentioned, is the one that's used, the principal one for messenger RNAs, and it's extremely important in that case to turn on or to turn off different genes. Real challenge to make sure that that genome of 30,000, most of that is specific genes. You've got to select amongst those. Polymerase 3 handles a number of the smaller RNAs, so it transcribes tRNAs and several families of small RNAs that we may or may not have time to talk about, but that are all a lot smaller than messenger RNA or ribosomal RNA. In addition, mitochondria and chloroplasts have polymerases. You may remember that we said that both those organelles had a little bit of DNA and that some part of the proteins inside those organelles was made inside that organelle. And so they can replicate their DNA and they can transcribe it. Mitochondria don't have a lot of genes there. Chloroplasts have rather a lot. But in both cases, the polymerases that are needed for that in part get things from the nucleus. And so they're very dependent on that relationship with the cytoplasm. And if we look at the structure of RNA polymerase 2, for instance, we might well see the affinities here to the DNA polymerase. We still have a palm, a thumb, and fingers, because it has to face basically the same challenge. The challenge of taking a linear piece of nucleic acid, selecting nucleotides to match it, and making a new bond. So the overall structure reflects the fact that the overall chemistry is fairly similar. Has some unique challenges, but a lot of the chemistry is the same. Like the DNA polymerase, it has magnesium. Its new chain is synthesized 5' to 3'. Like DNA polymerase, it has a template. Unlike DNA polymerase, it does not need a primer. So RNA polymerase can start in the middle of things and start a brand new, virgin, never been touched molecule of RNA. It can start things, whereas DNA polymerase is dependent upon there being a primer to work off of. So there is a brief period of time when the new RNA transcript is in a base paired hybrid with the DNA. And it's harking back again to our structural biochemistry. We mentioned that the bulk of DNA in our cells is in the B form, which is the sort of medium member of the trio. But an RNA-DNA hybrid structurally has some similarities to aDNA in terms of tilting of bases and things like that. So there's this kind of in-between state that it can happen. The DNA molecule is going to enter through two parts of the molecule called jaws, and they're going to function to separate it. and that's going to open up the transcription bubble. And then there's a polymerization site where the DNA has to sort of bend a bit to leave room for nucleotide triphosphates to come in for the polymerization reaction. The polymerase cannot select which nucleotide triphosphate comes in. So it's just letting in NTPs randomly without knowing what it's letting in. And whether or not there's a reaction or not will always depend on what the sequence is that NTP is encountering. This is just showing there's a slightly different way of making some polypeptides. This is a polypeptide antibiotic, which is not made by ribosomes, but instead by an enzyme complex where there's a separate enzyme for each peptide bond. That's obviously not something you can do for most proteins. It has to be limited to a very small number of peptides. Okay. RNA polymerase is also a very highly processive enzyme. You may remember that Carissa talked about how DNA polymerase gets on the DNA and goes for a long time. RNA polymerase has to be highly processive as well, because if its product isn't a full-length messenger RNA, the effort's all been wasted. And part of what does that is there is a clamp like this that can swivel around a little bit and kind of grab onto the DNA. So you see it's kind of like putting its elbow on top of the DNA to kind of keep it as part of the complex. And the interaction there is with a single-stranded DNA. When you think about the fact that genes can be, 1,000 to 1,000,000 nucleotides long, you can see that the RNA polymerase has to be very, very processive. Okay. So if we were to look at the sort of the overview view of how an RNA polymerase works, this is sort of what's going on here. There is an entry site for the entering DNA to enter. The region in which it's opening up into an actual bubble here. This part is going to be available as the template for the growing RNA strand. Here is the funnel that lets the nucleotide triphosphates enter and potentially hybridize or not, depending on what the base is that's there. The RNA that's growing has to leave, and the DNA has to reanneal back into a double helix. So that's what has to happen for everything to stay intact and yet allow enough movement of the DNA to be able to make new RNA against it. It's just sort of showing you the direction of movement. So. Interestingly, RNA polymerase can proofread. That this is not something that was appreciated until quite recently. And part of the reason it can proofread is because if there is an error, then in this part of the helix, it's the hybrid helix, the RNA-DNA helix, that error will distort the helix. And those distortions will then trigger the RNA to back out of that channel. It'll just literally back up, and then there's a nuclease that TF2S can stimulate to trim off that bit of the RNA, and then you can simply try over again and hope to do a better job the second time. This is showing you that from a slightly different perspective. Here you have the DNA entering and leaving. You have the growing RNA molecule here. As you elongate, an error is made. So the RNA error part has backed out of the funnel, gets cleaved off, and now you can try again at that particular part of the sequence and hope to do a better job the second time. So we've seen proofreading in DNA synthesis. We've seen proofreading in RNA synthesis. Can you guess if we're going to see proofreading in protein synthesis? In the early days of looking at transcription, people were sort of somehow expecting eukaryotic transcription to be similar to prokaryotic transcription and to sort of be an on or off kind of thing. You're either sitting there blocked by a repressor or you're zipping along making RNA. But that on-off character that prokaryotes have is not the way eukaryotes do it. Instead, at a promoter, you can detect many short molecules of RNA being made that then degrade very quickly. Only once in a while does it go on to make a long molecule of RNA. And so in the early stages, this complex can make a little bit of RNA and then may fall off again. To go from this preliminary complex where you have most of the factors together, but it's just making little bits of RNA, to elongate to where you have a complex that is competent to keep going processively for a long time. There is a region of the RNA polymerase called the CTD, the C-terminal domain. And this C-terminal domain has a sequence that's very rich in serines and threonines. 52 copies of that sequence. Some people, and so those are obviously targets for being phosphorylated. And if you think of that many serines being phosphorylated, you can see how the CTD got the nickname the negative noodle. That this is a piece of DNA that kind of flops out with a lot of negative charge on it and is now out of the way, can knock off complexes like the mediator. It can leave behind general transcription factors at the promoter so that the next round of initiation can go more quickly, but the competent elongation complex is now able to go very quickly down the template and make a long piece of RNA effectively. So what kind of a role might the histones play in all of this? We've talked about how histone acetyltransferases seem to be associated somehow with loosening the interaction of histones with DNA. We know that when we see histone acetylation, it tends to be in regions where the chromatin is somewhat decondensed. And we think it's going something kind of like this. So here we have an RNA polymerase rolling along, and it has to be able to go around that nucleosome without actually knocking that completely out of the way. And remember that there are two turns around a nucleosome. So the understanding is that there's room for polymerase to get started around it, that you could have enough of a loop for the polymerase to negotiate and still have contact with the rest of that part of the DNA to its nucleosome, and continues to wrap around, and it's come off the other side without that nucleosome ever being completely out of contact with the DNA. And you can imagine how if this nucleosome, these histones rather, have been acetylated, that it would be easier for these complexes to loop up and go around. So I think we're going to call that a day. I'll see you Friday. Yeah? Sure.
I was just noticing that we have both the second serine and the fifth serine phosphorylated for elongation. I thought that the fifth serine lost its phosphorylation, and then before it continued elongating, like it was initiated, it had to phosphorylate both to initiate, and then it lost the phosphate on the fifth serine to elongate.
I'm not, let me get this off.
Also, I was just curious. It's quite possible that that's the case. I don't know the fine details of this. Do you think that what we said here is misleading to people, or do you think that we should?
I was just hoping to double check to see just to clarify.
I would defer to that in terms of this. I'm not a specialist in RNA elongation. And so you're saying that which one does which?
Okay, to initiate both the phosphorylate, both the serines, and then the fifth serine loses its phosphates to elongate, and then and then like when the RNA stalls, they're both lost, and then it needs a new kinase to phosphorylate again to restart. So I was just curious about that.
Yeah. I think I'm going to leave it the way I said because I don't think I'm saying all of them are being
Right,
phosphorylated.
okay.
So I think that's a general enough statement to be consistent with what you're saying.
Okay.
Are you comfortable with
No,
that?
yes, that's
Yeah.
fine. I just wanted to cross-reference our
Well,
tests.
I'm happy to learn something, but I also don't want to confuse
Yeah,
this.
I didn't want to confuse anybody else.
No, thanks very much. No, thank you.